Hello everyone, welcome to Vivid Botany. We have one more question from same chapter molecular basis of inheritance. This question is based on the RNA polymerase types in eukaryotes. See, I was telling you in my previous question also that the eukaryotes have two particular phenomena that are different from prokaryotes. What they are? Eukaryotes have two specific phenomena because it is very complicated, very complex. Let us see what they are. The two specific phenomena include here. One is split gene arrangement. I told you split gene arrangement means having introns and exons is called as split gene arrangement, which are present only in eukaryotes but not in prokaryotes. Another is division of labor. Division of labor in RNA polymerase. Division of labor in RNA polymerase. That is, there are three kinds of RNA polymerase are there. Division of labor means according to that they form the RNA polymerase can be divided into three types. There are three types of RNA polymerases. What they are? They are simply named as RNA polymerase 1. So this RNA polymerase 1 will, RNA polymerase means remember it is an enzyme. An enzyme is made up of proteins. So this enzyme will synthesize rRNAs, that are ribosomal RNAs. What are these rRNAs? They include here 28S rRNA, 18S rRNA and 5.8S rRNA. These three rRNAs are synthesized from RNA polymerase 1. I think you know what is S. S stands for Swedberg unit. This is a parameter of the ribosomes during the sedimentation, during the centrifugation process. During centrifugation, ribosomes will undergo sedimentation due to their density, and that is measured in terms of Swedberg units. And here they are 28S, 18S, 5.8S. RNA polymerase 2 will synthesize here what we call as I have told you in my previous question also, that is hnRNA. hnRNA stands for heterogeneous nuclear RNA. Why it is called as heterogeneous nucleus RNA? Because it contains the functional genes, non-functional genes. That is, functional genes are called as exons, non-functional genes are called as introns. So we call it as heterogeneous nuclear RNA or hnRNA, or we call it as precursor of mRNA. Precursor of mRNA means mRNA is formed from this heterogeneous nuclear RNA. We have the term precursor, I think somewhere, that is what is the precursor of auxins? Auxin precursor is tryptophan and zinc. Tryptophan is an essential amino acid and zinc is required for synthesis of tryptophan. So tryptophan, zinc, both are precursors of the auxins. And similarly, precursor of mRNA is hnRNA. This hnRNA is formed from RNA polymerase 2. Let us see what is RNA polymerase 3. RNA polymerase 3 synthesizes three molecules that include here. One is tRNA, that is transfer RNA. Remember, it is the first discovered RNA. It's called as soluble RNA. It's called smallest RNA. Since it carries the amino acids to the site of protein synthesis, it's also called as adapter RNA. So these tRNAs are 61 types because sensible codons are 61. So for 61 sensible codons we find 61 types of the tRNAs. So this tRNA is synthesized from so-called RNA polymerase 3. Apart from that, RNA polymerase 3 synthesizes 5S rRNA. See, the 28S, 18S, 5.8S rRNAs are synthesized from RNA polymerase 1, whereas 5S rRNA synthesized from the RNA polymerase 3. For example, a question can be framed that find out the odd rRNA among the following, that include option 1 28S, option 2 18S, option 3 5.8 yes, and option 4 5S. Option 4 is the right answer. Why option 4 is the right answer? Because the first 3 rRNAs are synthesized from RNA polymerase 1, whereas 5S rRNA synthesized from RNA polymerase 3. So like that, a question can be framed here. And one more, that is snRNA. snRNA stands for small nuclear RNA. So small nuclear RNA is meant for splicing. Do remember, it is meant for splicing. Splicing means removal of introns, and removal of introns is called as splicing. This splicing is done by small nuclear RNA, and this small nuclear RNA is formed from RNA polymerase 3. So this is what I have described as division of labor of RNA polymerase. And three kinds of RNA polymerases will show three different functions only in eukaryotes. Remember, eukaryotes have split gene arrangement, eukaryotes have the splicing, processing of mRNA, and eukaryotes have three types of RNA polymerases: RNA polymerase 1, RNA polymerase 2, RNA polymerase 3. Now, let's see the question. What is the role of RNA polymerase 3 in the process of transcription in eukaryotes? He is asking here RNA polymerase 3. So it transcribes rRNAs 28S, 18S, 5.8. It is wrong because these are transcribed by RNA polymerase 1. RNA polymerase 1. Next question. It transcribes tRNA, 5sRNA, snRNA. This is right. Directly it is taken. Third option, transcribes precursor of mRNA. No, precursor of mRNA is by RNA polymerase 2. So this is also wrong. Transcribes snRNA. Only he is saying, you see, only snRNA is wrong because apart from snRNA here, tRNA, 5sRNA are also synthesized here. So fourth option is also wrong. The correct answer is here option 2. So this is very peculiar phenomena seen in eukaryotes, that one should remember the division of labor and the functionality of RNA polymerase 1, 2, 3, as well as this split gene arrangement seen in the eukaryotes. So he is asking here directly what are the functions of 1 and 2, 3, and the previous question also he asked split gene arrangement, splicing, capping, tailing. So very directly lines are taken from the textbook. So one should read them very thoroughly. Once again I am requesting you to draw the diagrams wherever you get, and label the diagrams, underline the keywords, make a separate notes for the keywords, for the concepts, and for the understandings. Then you can do better. And I think with this preparation, definitely you are going to have 180 out of 180 in what do you call this botany. I mean, I am taking of only botany, I am taking 180 out 180, and definitely you will get a victorious end at the NEET question paper, NEET exam. So hope my video is helpful for you, and do not forget to subscribe the channel. We will be counter attacking each and every question in detail. I'll be discussing each and every term in detail so that you can concretize your concepts, you can become more confident, you can have more booster from the discussion. So let us don't forget to subscribe my channel Vivid Botany. We will be meeting in our next question. Thank you.